What's up, Matrix? It's finally your time to shine. You hang out with Looney, and then he said the mad guy is back. Aslam, how are you?
Looney, what's happening? They call you Looney, but I'm the mad guy.
Yeah, you know,
That's the problem, eh?
this thing. What are we doing for the great house today?
Today we're doing the magic stuff, DNA, that thing that gives you your color of your eyes and the shape of your eyebrows and the rest of it.
Everything. Okay. Great House, I hope you guys are excited. Just before we start, happy birthday to Llewellyn, who didn't tell us it was his birthday today. Happy birthday to you. Hope you enjoy the rest of your day. And mindset is to get a hold of us, Facebook and Twitter, @LearnExtra. And you can visit our website, learn.mindset.co.za. And then Aslam will give you a great lesson.
Wow, Looney, you're learning to talk a bit faster.
You know, I'm trying. I'm trying. It's a new year. It's a new year.
Hi, guys, and welcome 2014 Grade 12s. This is your big year. The year where the rest of your life may be decided in terms of a career or job or whatever else that you're thinking about doing. Take it very seriously, start working from the beginning, and we shouldn't have any problems. Today we're looking at DNA, deoxyribose nucleic acid, and this is one part or one component of nucleic acids. We'll come to that just now. What do we want to really do today? The idea we put this on, guys, is at the end of this hour, these are the things that you need to know. So after the lesson, you need to ask yourself, do I meet these things? Okay? So the first thing we want to do is revise the structure of the cell with an emphasis of the ribosome, cytoplasm, and parts of the nucleus. This is what you would do. You will revise the cell. Go into the cell, look at the nucleus, look at the ribosome and the cytoplasm. Most importantly is the nucleus in this whole topic that we are talking about. Name the two types of nucleic acid. And again, while we're talking about this, guys, remember, look at the words. If this was a question, it said name. Did you have to discuss or explain anything? No. Name the two types. So you're going to just say what they are. We'll talk about that more later. State, again, another verb, saying state where it occurs. So there's no discussion. You just got to give the location. Describe. I'm so glad. And guys, I must tell you, this is not even planned. And I'm so glad that it was put this way, because these verbs are the most important thing in the life science. The verbs in the question paper I'm talking about. Just today I was at a roadshow and we were talking about the action verbs. But we'll talk as we go along. Look at this one. It says describe now. Describe the history of the discovery of the DNA molecule, Watson, Crick, Franklin story. What happened? How was this molecule discovered, etc. Again, describe the structure and name. Look at that now. In the one sentence, you're getting two action verbs. Describe the structure. In other words, you're going to tell us what the structure is, and name the three components of a DNA nucleotide. A big word there as well. We'll talk about that a little bit later. As follows. State the functions of DNA and describe DNA replication. The three components missing here for some reason is the phosphate, the deoxyribose sugar, and the nitrogenous base. These three things make up a nucleotide. But we're going to go into details just now. Then you must know the functions of DNA. Why do we have DNA in the body? And also describe DNA replication. We must also be able to talk about DNA fingerprinting somewhere in the show as well. Now, you would know that the way we've designed the program now, we have changed the way we do the lessons. So if you were in grade 11 and you were following last year, you'd know that we were doing a lot of content and so on. Now we're moving away from that. We are trying to hone in the skills, and the answers to the questions should be your content. So before we even start with today's show, we have a challenge question, which it's already, the challenge question, Looney, is already on Facebook, yes?
Yes.
It's already on the page. And I'll just read it for you again. A DNA molecule has 2,000 bases. What percentage of adenine bases is present if 400 of the bases are cytosine? That's the first part of the question. The second part of the question, what will be the percentage of guanine in this case? In the same case, what will be the percentage of guanine? Hint, remember, in DNA, the way the nitrogenous bases are combined, we have at cute girl. At adenine combines with thymine, and cytosine combines with guanine. So we're talking about at cute girl. That's how you're going to remember that A combines with T and C combines with G in DNA. So you think about that, you answer that question, and try and show working when you're answering the question so it makes sense to you. Now let's look at what this section is all about. Summary of the work that we're going to do. What are we talking about? DNA. But in this DNA, we don't only look at DNA itself, but today we're looking at DNA, remember that. But on the whole, the section deals with nucleic acids. These are molecules that are found in the nucleus and the cytoplasm. What are these two nucleic acids that we are talking about? We talk about DNA, deoxyribose nucleic acid. The SE normally is silent, deoxyribonucleic acid, and ribose, RNA, ribonucleic acid. Two important nucleotides that we find in the body. We are going to just look at, first of all, where we find them. DNA is found in the nucleus, and some DNA is found in the mitochondria. In grade 11 or in grade 10, you may even have learned that some DNA is also found in the chloroplast. For our studies in grade 12, we are worried about these two, the one in the nucleus and the one in the mitochondria. Whereas RNA is found in the nucleus and in the cytoplasm. So RNA is also found in the nucleus and it's also found in the cytoplasm. Today we're not going to go into too much of detail about RNA. We are more interested in the DNA that we are looking at here. What do we need to know about this DNA? We need to know, right, the history of the DNA, and we also need to know the structure of the DNA. Further, we must also look at the DNA replication, and finally we look at DNA profiling. Now if we look at each of these things, what do we need to know? When we're talking about a history, we want to know about the discovery of DNA. And here we talk about 4 scientists. We talk about Watson, James Watson, and we talk about Crick, Francis Crick. We talk about Franklin, and we talk about Wilkins, that is Rosalind Franklin and Maurice Wilkins. These are the 4 scientists that are somehow involved in the discovery of DNA the way that we know it today. In other words, they have broken the way forward to tell us of how DNA looks. In other words, when we're describing DNA today, we owe it to them. Now, in your on the notes that you have probably downloaded already, you'll find a series of multiple choice questions that go through these. As you answer those questions, it gives you an idea about how this unfolded, the structure of the DNA unfolded. Okay? Then we look at the structure of the DNA. And what do we need you to know there? We need you to know, firstly, that DNA is a double-stranded molecule, double strand. And this double strand is spiral, so it's a helical structure. Helix. Spiral. We also need you to know that DNA is made up of the nucleotides, and the nucleotides are made up of phosphate, as I said earlier, phosphate, sugar, and in this case, which sugar? Deoxyribo. and a nitrogenous base. These nucleotides, in other words, we have a P, an S, and a nitrogenous base. Each one like this represents a building block of the big molecule. DNA is a large molecule. Where is this DNA found? This DNA is found in the nucleus. Where exactly? In the nucleus we find chromosomes. The chromosomes are made up of chromatids or chromatids. What's the spelling? That's a D. Chromatids. And the chromatids, each one is made up of long strands of DNA. So this is where we find the DNA in the cell. This is all that you've already learned in class. Okay? In terms of replication, we're not going to talk much here because we have a question based on that, and that question will take care of that section. But what is replication? Simply making copies. DNA replication refers to that process where DNA makes a copy of itself. Why, how, etc. we will answer just now. And the last part of this work deals with DNA profiling. DNA profiling is a technique where individuals are identified by their patterns of the DNA in their body. And, you know, 10, 15 years ago, or 25 years ago, or the time when I was in school in grade 12 or grade 11 when we did DNA, if somebody told me about DNA profiling, I would have scratched my head. Because this technique of DNA profiling, for a start, was only really discovered in 1986 in the UK, in Leicester University. A gentleman used that to show how we can match DNA to a person. But today, Luni, today, we hear about DNA profiling every day. You switch any channel, medical detectives, CSI, NCIS, yes. Every crime that they're talking about, they want to match the DNA. And they, at an advanced state, in the States, they are in an advanced stage in this profiling. We are still way behind because it costs money, the equipment is expensive, and so on and so on and so on. But there they are in such a case where they have DNA profiles of certain criminals and also mainly of the soldiers. All the soldiers in America have a DNA profile, and the DNA profile is in a computer. So if they find, for example, remember they are in Afghanistan, they may be somewhere else, and loads of them are getting killed on a daily basis. And if that charred remains, the burnt out remains of the body comes there, and they need to identify it. They extract the DNA, and from there, they will then match it on the computer to say that this is Luni or this is Aslam. If this building for some reason burns out and nobody can recognize us, but they probably recognize me with my grey beard. I don't know about Luni. But with me, they check a couple of grey hairs lying around me. They'll know that that was the mad scientist or the mad Khan biology guy. But in terms of Luni, it would be harder to recognize. You know, she's young, and there's so many young people out there. So we are past the age, so there's not many old people in the building at the moment. So in that case, this is why we use DNA profiling. Where do we get this DNA from? DNA is found in every cell in your body, because every cell has nucleus, and every nucleus will have chromosomes, and therefore every chromosome will have DNA. So in every cell, if you cut your nails and you take that nail sample, the scientists can extract DNA from your nail sample and analyze it. Much like, now that's why I left this for last, this last part that I'm going to talk, because if I said it first, then you would have used this in exams and you will get no marks for it. Much like if you're buying an item in the store, you buy lunch bar, Luni, you go to the till. How come the computer knows that's lunch bar? Does the computer have eyes? It has a barcode. Good. So each item there has a barcode. So you and I, we have our own barcode. And with that, Luni?
All right. Mindset is, we are going to take a break, but before you do, Aslam, a special shout out for you from Belinda and Letitia from out there in the world. They say they miss you. And hi, hope you're doing well.
Thank you so much.
Mindset is, we are going to take a very short break, so do stay tuned and I'll see you straight after this. Welcome back, Mindsetters. From that short break, I hope you guys are nice and energized and you're ready to take in some more information from Aslam. Just a message from Aslam. Guys, some of the questions that you are asking on Facebook will be answered now in this segment because he's going through more information. So you're asking like questions that he has prepared for you guys. So, Aslam.
Thanks, Lune. Guys, just another thing. Just remember that you've already started school 2 weeks ago. Almost 3 weeks actually. So you've already covered this working class. So our purpose is to guide you through, and remember at grade 12, the content you have to still learn obviously, but more importantly is the skills of answering questions. So while you getting your lessons in school also, get hold of last year's paper, get hold of last year's trial paper and a couple of other papers prior to that. Just remember that once you do a section, for example, you do DNA at school, you answer some questions on DNA on your own. So you keep ahead and you keep abreast with what's happening. Study the diagram. Going now to the questions. The question 1 says, study the diagram below, which represents a part of a nucleic acid a molecule, and answer the questions that follow. Now, obviously, in today's show, this question will be easier than at the end of the year, because you have only learned one nucleic acid so far. And by the end of the year, or even by next week already, you would have learned the other nucleic acid, and you may have learned other molecules as well. Now it becomes confusing. You've got to choose from those different molecules. But today it's easier. But this is a typical exam type question. So you need to get familiar with this type of work to keep yourself on the toes. Right. So they're giving you a diagram, and they label they're given numbers there, and they give you a key as well. And the key says A is adenine. And obviously G is guanine. First of all, they say identify the nucleic acid shown in the diagram above. And we've just done this work, so this has to be G and A. Remember, you don't have to write out in full deoxyribonucleic acid. DNA is an accepted abbreviation. Okay? Now, I can ask you another question before we even look at the next question here. After you've learned both molecules, another question may ask you, give a reason for your answer. In other words, why are you saying that this is DNA? In this particular diagram, there's only one proof at the moment, and that is that it has 2 strands. So it's double-stranded. This is not part of this answer, remember. I'm only giving this as an extension to this question. If they ask you, how do you say that this is DNA? Okay? And you've already done RNA, so you understand what I'm saying. Now they're saying label the following: part 1, part 2, and nitrogenous base 3. So part 1 is pointing to this guy here, part 2 to that guy there, and then they ask you for nitrogenous base number 3 there. Okay? Easy stuff. We do it on the diagram. So part 1 is the round one. Remember the outermost part of this structure, the backbone, the side bones there, would be phosphate. Number 2 would be the deoxyribose sugar. I'm just writing the sugar in large so you understand what I'm saying. But remember, whenever there's DNA, you can't just say sugar, you must say deoxyribose sugar. When it's RNA, you must say it's a ribose sugar. Otherwise, you will not get the mark. And nitrogenous base number 3. Now, what is this here? What letter do you see there? The letter A. And this said that this is adenine. Therefore, I said at cute girl. So adenine will combine with thymine. So our answer there would be T or thymine. You write it in full, thymine. What is the collective name for the parts numbered 1, 2, and 3? Let's go back. In other words, this one here, I'm going to use another color there for you. Let's do that. So we're talking about that part there. 1, 2, sorry, not that part. Let's just undo that. Okay. So we're talking about there, 1, 2, and 3. There you go. I said to you earlier, the name for that is called a nucleotide. Nucleotide. Good. Are we together, guys? I'm hoping we're together. Comment on the significance of the nature of the bond labeled for. I love this question. I love to ask this question. Comment on the significance of the nature of the bond labeled for. What do we mean by significance? Significance means, why is it there? What's so good about having this type of bond? So you can't comment on the significance if you don't know the nature of the bond. Let's go back to our diagram. The bond labeled for, not James Bond 007, the bond labeled for. Okay, oops, for some reason the label line is in the wrong place, so it should be labeled 5 in this case here. So we're talking about the bonds there, all these bonds, the bonds between the nitrogenous bases. Okay? So comment on the significance of that. So what do we know about these bonds? What bonds are they? They are weak hydrogen bonds. So what's so good about having weak hydrogen bonds? They break easily to allow for replication. Simple. Look at the question. If it's standing there staring at you, comment on the significance of the nature of the bonds labeled so and so. There's only one bond in DNA molecule that we talk about in really for our study purposes, and that is the weak hydrogen bond. And the word weak is important there. The fact that it's weak, it is easy to break this bond when replication takes place. That means when this double strand separates into 2, and then it can reform again after replication. I'm hoping we're all on the same page there. Okay? The next question says, the diagram below shows part of a DNA molecule in a nucleus just before cell division. Okay? Just before cell division. And they've given you a key there and whatever. But you can see what's happening here. Something is happening that this molecule is splitting. So identify the parts labeled 3 and 4. Notice the overlap now from what we just did. So in this question, although it's dealing with something else, but it's still dealing with the structure as well. 3 and 4. Let's go to 3 and 4. Okay, there's our 3 there. That would be the deoxyribose sugar. And 4, there is the phosphate. Identify the nitrogenous bases labeled 1 and 2. Whenever they're asking you nitrogenous base, they are testing one simple thing. Do you know that adenine combines with thymine? Do you know that cytosine combines with guanine? That's all they're testing you. So that's what you've got to look for. So number 1, number 2. Let's go to the diagram. Now let's look at the strand. This one was a little bit complicated. If this is the strand and it starts there, then obviously that one combines there, 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 etc., and there. Okay? That's how the molecule was originally. So what was on the other side of number 1 and number 2? Let's look for number 1. Number 1 is this one here. This one here is number 1, there. So where did it come from? It came from that, it was combined originally to that guy. So nitrogenous base number 1 would have to be CG. So it's guanine. guanine with the G, no? Okay, number 2, here's number 2. Number 2 was originally, no, look at where number 2 is, right? This is the catch one. Look at where number 2 is. The number 2 is this one here. But number 2 is combining with this guy here. And this guy here was originally combined to C, so it was G. So therefore, number 2, sorry, has to be again another, would be cytosine, obviously, because this is G. So this will be cytosine. C. Let's see the logic behind that again. This particular nitrogenous base here was combined to, where am I going? I'm losing my mind here. Let's just undo that before I get carried away there. Okay, so this one here I said was combined with this guy here. Okay, so that was G. So sorry, we've got to go back. And so this will have to be C. Sorry, let me change my answer there. This would be C, not G. So this must be G. Again, another guanine. So we take that out, that becomes guanine. Sorry about that. I looked at the nitrogenous base above. Now remember, you need to match those bases, and you would have counted 1, 2, 3, 4, and will make it the match that way as well. Okay? Explain why the diagram above represents replication and not transcription. Okay? Why does it represent replication and not transcription? What is happening? What base are you getting here? Thymine. Okay? Thymine. And because of that, this will show that this is the replication and not transcription, because in transcription involves a messenger RNA, and messenger RNA would have uracil and not thymine. Okay? So because the nucleotide forming, it has thymine. Therefore, it cannot represent messenger RNA. Okay? Question 3. This is a longer question now. They give you a case study, and they're saying you must answer the question. This guy was investigating the scene of a violent crime. 70-year-old woman. She was stabbed and left to die. They found a few pieces of hair in one of her hands. There was also skin under her nails. They arrested 3 possible suspects. DNA samples were taken from all 3, and hence we come to this point here. So here we're getting the DNA sample from the hair. Here. There's the DNA sample from the victim, suspect 1, 2, and 3. Identify the suspect responsible for the crime, carrying out the crime that is above, in the above evidence there, right? Now, how do we do this? This is a simple matching thing. If we found evidence on the crime scene, and if the suspect has the same DNA, then it's probably him or her. Okay? So what do? We look at this DNA sample. Is it exactly like this one? No. Is it exactly like that one? Already on the top, it's wrong. Is it exactly like that one? No. Is it exactly as that one there? Yes, it does. So this one here is the suspect, suspect 3. And you'll notice whenever they give you a question like this, they always follow it up with provide a reason for your answer above. No matter which case scenario you get, your answer for the second one will always be the same. Why? Because suspect, whichever suspect you choose, because suspect 3 DNA is identical to the DNA of the hair found at the scene. As you're going to grow into life science, you're going to realize that these type of questions are asked and the answer is always the same. Suggest a reason for the comparing the victim's DNA with the other samples. Oh, we got a hair, we got the suspects. Why must we take the victims? We take the victim's hair because we want to compare the sample with the victim, so that we know that the victim's The hair doesn't belong to the victim. Otherwise, we look for a criminal till we die. We won't find them because we would have buried the criminal, the victim. And the last one, do you think that DNA evidence on its own is enough to convict a suspect? No, obviously not, because the hair could have been planted there on the scene. Somebody could have framed this person. There could be mistakes in the lab, and this person could have an identical twin. With that, Looney.
Mindsetters, let's take a quick break and we'll be back with more questions straight after this. Welcome back, Mindsetters, from that break. I hope you guys are still enjoying the show. Where I can see on the Facebook page you are, because you're answering questions, you're asking questions. So right now, Aslam, we are going to do the challenge question. Okay, so Mindsetters, it is posted on the page, and a lot of you have tried to work it out. So Aslam is going to help us with that now, and then we'll go through the Facebook questions.
Good. Great, Looney.
Yes.
Guys, I'm hoping you're following what we're saying, and I'm hoping many of you tried this question out. And now is the time to reward yourself if you got it right. This is why it's called a challenge question. You need to tap yourself, hey, yeah, I got it right. This is one of the more difficult ones in DNA that they like to ask. Instead of just telling you match the A with the T and the G with the C, this question is doing the same thing. Only thing is making it a mathematical problem. So again, our basis of this question is based on the fact that the number of A is equal to the number of T and the number of C is equal to the number of G, because of base pairing. Let's write that so you won't forget it. Base pairing. For every A in the DNA molecule, there will be a T. And for every C, there will be G. Why? Because DNA is a double strand. And why? Because A combines with T and G combines with C. So if we have two strands and the one has A, T, G, T, G, 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 C, 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 then the other one must have the opposite number, a member of the group. So they're saying here a DNA molecule, and let's now learn the other skill of underlining keywords. A DNA molecule has 2,000 bases. Important to know that. Altogether it has 2,000 bases. What percentage? So that already tells you the unit that you're going to give in your answer. Your answer has to be in percentage. Of adenine bases is present if 400 of the bases are cytosine. Adenine, cytosine, but they don't combine. Damn, these guys are making our life difficult, eh? What the hell, man? Why are they making our life difficult? Okay? That's what you're thinking right now, but it's so simple. First of all, if 400 bases are cytosine, we need to work out, first of all, if 400 bases are cytosine, what? Because we're going to work in percentage, not in number. So we need to know what percentage is 400 of 2,000. Okay? So, for your OBE lot, we're going to say 2,000 divided by, or rather, sorry, what am I doing here? Now I'm doing something very wrong. We're talking about 400 bases. Now watch why we're working it out in percentage. We have 400. If you got a test and you got 20 out of 50, how do you work out your percentage? You say 20 over 50 times 100 to give you a percentage. You're going to use your calculator. You're not going to do all of that. You're going to press the percentage button. So 20 over 50 times 100, in this case it's very easy, is equal to 40%. Okay? So that's what we're doing here. We got 400 out of 2,000. So we're saying 400 divided by 2,000 out of 2,000. And equal to, it's 1/5 obviously. 1/5. How many percent is that? That is 20%. Okay, equal to 20%. Now, if that is what cytosine is worth to this particular calculation, what do we know about cytosine? For the same amount must be guanine. So guanine, so now we're saying, we worked that out. We said, what did you say? 400 divided by 2000 times 100 over 1. And it gives us 20% for C. And because of base pairing, then G is also equal to 20%. Why? Because for every cytosine there must be a guanine. All right? But they didn't ask you for all this. They asked you for adenine. Now why are we working all that? We have to work that out to work out adenine. So adenine, if we want to work it out, we know now we got 40% here. Adenine is equals to 100% minus 40%. Let's take away that brackets there. Okay, we took everything out. Minus 40%, come on. Minus 40%. Okay. Divided by 2. Hey, what the hell, man? This is life science. Why are we doing some heavy maths here? Let's understand what we're saying here. 100 minus 40, we're getting rid of that. It's equal to 60%, obviously, isn't it? Why are we dividing that by 2? Because we have 2 more nitrogenous bases to account for, and that is adenine and thymine. And they are also equal. So we divide it by 2, and therefore the answer is 30% is adenine. The second question asks you, what will be the percentage of guanine? We've already worked that out. Because of base pairing, guanine is 20%. So the last question was just testing to see whether you understand the concept of base pairing. In fact, the whole question is based on 2 things. One is a little bit of calculation skill, but more importantly, do you know that when you're doing this work, that for every adenine there must be a thymine, and for every cytosine there must be a guanine. And that's what we got. What are our guys saying there? Are they saying they got it right? Are they happy with the answer, Luni?
Yes.
They are? Okay, let's take their questions now.
Okay, cool. Have quite a lot. Okay, from Semakeleng Motseme. I think it's a he.
Yeah.
Enzymes are proteins, and proteins are products of DNA after protein synthesis. So how can enzymes dominate as catalysts in most processes of DNA?
I'm going to have to say that one slowly.
Yes.
Enzymes are proteins.
Okay, let's start from the beginning. Enzymes
are proteins. Let me just jot as we go along.
Okay.
Enzymes
are proteins.
Are proteins.
And
good.
Proteins are
the
products.
Others, by the way, we're writing it so you can follow the question, right?
Okay.
And proteins.
And proteins are products of DNA after protein synthesis. Yes. So now the question is, how can enzymes dominate as catalysts in most processes of DNA?
Ah, I see the question.
Yes.
Your question is at a very high level in terms of the language you've used, and you're going to confuse a lot of people. What the youngster wants to know is that enzymes are proteins. And you've learned protein synthesis, and you know that DNA is involved in making the protein. DNA gives the code for protein synthesis to take place. Yeah? Okay? The youngster wants to know, how can the enzyme dominate reactions involving DNA when, in fact, DNA produces the enzyme? Yes, young man, this is beyond the syllabus, but nonetheless, let's give you an answer for that. Why it's not dominating the DNA as such, what it's doing in DNA replication and in transcription and translation, all the enzyme does, it catalyzes the reaction that takes place. What do we mean? It speeds up the reaction that's going to take place. In other words, it lowers, grade 10 work now, it lowers the activation energy required. Now, I'm talking a lot of stuff here. Let's bring it down to a very simple thing. What energy do we require to boil water? We need 100 degrees Celsius of heat to get water to boil. All right? So that's the amount of energy that we need to get the water to boil. Now, in our cells, we have various reactions taking place, including the fact that the DNA must split and the DNA must recombine and so on. For these reactions to take place, requires a certain amount of energy. And if we had to carry out these reactions at that high level of energy required, we would probably burn out because the energy requirement would have been too high. The enzyme's job is not to dominate, but it simply comes in there to bring the activation energy lower so that this reaction can take place at a faster rate. That's all the enzyme does there. Okay?
Cool, thanks. And then Seth2+ is asking, what is the definition of the nucleic acid?
What is the definition of a nucleic acid? There's no definition of nucleic acid. Suffice to say that nucleic acids are macromolecules or polymers, very big molecules that are found in the nucleus and in the cytoplasm. There are two types that we study, DNA and RNA.
Cool. Brighton Swing is asking, what is the function of the mitochondrial DNA?
Give me the name again.
Bright.
Bright. Like in bright.
Bright.
Okay. Bright wants to know what is the function of mitochondrial DNA, and we haven't covered it in our question, so I'm glad they asked that question. The mitochondrial DNA, by the way, the mitochondrial DNA, also called mtDNA. First of all, where is it found? It's found in the mitochondria, obvious. This mitochondrial DNA is used by geneticists to show relationships between individuals. And I'm giving you the relation to our syllabus. Remember, we can go into very detailed functions of the mitochondrial DNA itself, but that is not required in a syllabus. What I'm giving you is what is its role in our syllabus. Because when you get, let's give you a simple example. Last year in paper 2, they were asked to draw the motor neuron. The labels that are prescribed in the syllabus are simply the cell membrane, the nucleus, the axon, the dendrite, and the myelin sheath. Those 5 are prescribed. There are also other labels on the motor neuron. That is the nodes of Ranvier and Nissl granules. If the learner gave those other labels, which are correct, Luni, but they are not prescribed, we did not mark that correct. We only gave marks for those things that are in the syllabus. Okay? So I'm going to give you an explanation in terms of the syllabus. The mitochondrial DNA is used by geneticists to show relationships between individuals. Now, how does it actually work? The mitochondrial DNA is passed from mother to child. Why? Because the sperm's contribution in fertilization is the nucleus of the sperm. That means the chromosome and the chromatin network that will fuse with the egg cell. The egg cell is a whole cell, and this egg cell will have mitochondria, etc. So the mitochondria that Luni has, she got from her mother. The mitochondria that I have, I got from my mother. So through this mitochondrial DNA, scientists are able to show ancestry from the maternal end. So we can go back to the maternal end. And by the way, evolutionists use this mitochondrial DNA to fight for or give evidence for the out of Africa hypothesis, because such mitochondrial DNA has been traced back to a female in Africa known as African Eve, and therefore they say that the Homo species arose in Africa. So that's why we talk about the mitochondrial DNA.
Okay, cool. And then Gabza is asking, why do you say suspect number 3 but you tick number 4?
Go back. What's the name of the person?
Cabello.
Cabello.
Yes.
We tick number 4. Cabello is he sure about that?
I'm sure he's sure if you ask the question.
Remember, Cabello, when I open it and if it's not number 3, I want you to turn around and kick yourself, okay? I'm hoping you can get that right, but I want you to kick yourself hard, okay?
Okay, so he says, you said it's suspect number 3, but you ticked number 4.
Okay, now where did I tick number 4, Luni?
Okay.
It's the fourth
I think
bar.
yes,
It is
that's
the
where
fourth
he's
bar,
confused.
Cabello.
Yes.
You must read. Now, the same way you must read in your question paper too, because these are the mistakes we make. I'm glad. Actually, I appreciate mistakes like these, because this helps us to explain that in an exam, sometimes they say, write down the letters and you give the name. You get 0, nothing. Okay, so Cabello, look properly. We circle number 3. It is the fourth bar. The first set, this one here, is the sample, the hair that we found at the scene. We took its DNA bar graph. It's barcode, yeah. Not bar graph, the barcode of this DNA, if you want to call, use the. The second bar is of the victim. That means the person that's dead there, we took their DNA. And 1, 2, 3. Those are the suspects. So therefore, it was suspect number 3. Good.
Okay.
But
and
good
then
question, Cabello. You're watching, that means. I'm happy about it.
thanks, Kabel. So, Kani Butelez is asking, so are there any other bonds excluding weak hydrogen bonds?
Between the two molecules, no. Between two amino acids, as we learn later, we'll talk of peptide bonds. You have spoken about ester links in fats in grade 10. There are many bonds in the body, and everywhere there will be bonds. But in DNA, we only talk of the weak hydrogen bonds between those things. Remember, there are also bonds between the phosphate and the sugar and the nitrogenous base. But we're not defining those bonds here. We are talking about the bonds between the two nitrogenous bases that keep the two strands together.
Okay. And then Tokozani Morgan is asking, is phosphate also called an ion?
Phosphate ion, yes, phosphate ion.
Ion.
It's fine. Remember, you cannot, I'm glad he also asked that question. Lots of learners talk about phosphorus. That's wrong. It has to be phosphate because it's a compound of phosphorus, right? Phosphate. Okay?
And then Nekavambe Kodani, is it true that me and my twin sis have the same DNA?
Twin sis?
Yes.
Is he a male or a female?
Female?
Both of them are female.
Female.
Right. What's the name? Nekovambe.
Yes.
I'm getting good, eh?
Trying.
Nekovambe. If your sister is your identical twin, it doesn't mean she's a girl and you're a girl that you're identical. You could be fraternal twins. Now let's just do a bit of history there. Identical twins arise from the same egg and the same sperm. In other words, fertilization takes place. One sperm fertilizes an egg, and as this egg develops, it splits into two. So obviously, you got the same chromosome from the father and the mother, from that particular sperm and that particular egg. Therefore, you will have the same DNA. You'll have identical DNA fingerprint. However, if you are fraternal twins, that means you have arisen from a different egg and a different sperm. That means two sperms fertilize two different eggs at the same time or in the time of fertility in your mother. And therefore, you may have been born as fraternal twins. If you are fraternal twins, then you will have, obviously, different DNA fingerprints because you came from different eggs and different sperms.
Cool. Rebaleni Nemutanzela.
I'm happy that you're asking the questions, guys. For us, the value is in your questions coming in this last session for your time.
Oh, it's Rabeleni.
Rabeleni.
Can we find DNA in our hair?
Definitely Rabeleni. If we take a sample of your hair and we extract the DNA from there, definitely we would find DNA. Every living cell in your body has DNA.
Okay. Offense at Daphne, how exactly am I supposed to write an essay to achieve more marks? Because I have noticed that I don't get much marks like I expect, though I include every specific word. Is it the flow of my essay? How do I achieve my skill?
What's the name again?
Offense.
Offense. You want to know how to write an essay? Very simple. It's a very good question as well because essay writing is important. Many learners lose their A or distinction because their essay is not up to scratch. First and foremost, my first advice is answer the question. Let's take the work we've done today. Let's take the work we've done today and let's make an essay out of it. just to show you that any section can be asked in an essay. So the question is describe the structure of DNA and explain how DNA makes a copy of itself. Okay, so there's the question. First and foremost, you identify the verbs here. So let's identify the verbs. The first one, describe. The second one, explain. Okay? Whatever comes after the verb is what you must do. Describe what? The structure of DNA. And explain what? How DNA makes a copy of itself. That big sentence there, how DNA makes a copy of itself, what does it talk about? It talks about replication. So two things you have to describe. Two things you have to do. You have to describe the structure and you have to talk about replication. Now, did the question ask you about the significances of DNA replication? No. Can you put that in your answer? No. Did it ask you where do we find DNA? No. Can you put in your answer? No. The minute you put those things in, that means you have in your answer some irrelevant information. So you're going to lose marks in synthesis. The way the synthesis is marked now has changed into R, L, and C. R for relevance, logical flow, and finally comprehensiveness. I'll just write comprehensive. In other words, relevance means whatever you've written in an essay, is it relevant to the question being asked? If not, you lose 1 mark. This is 1 mark, 1 mark, 1 mark. Okay, I'm not talking about the content now. Logical flow. Did you start talking about the structure of DNA and in between a sentence you said when DNA replication takes place, something happens, then you come back to the structure, then you go back to DNA replication, so there's no logical flow? Or did you explain the structure first and then you went to DNA replication? If you've done it that way, then you got logical flow. And did you answer both parts of the question? That means you've spoken about the structure and you've spoken about DNA replication. Then you get full marks for it. However, now let's go back to the theory. When you're answering this question, you must only include information about the structure and replication in that case. So your content is then covered in that work. And you have answered the question comprehensively.
Okay. This is from Tapedi. Is there any chance that thymine will pair with cytosine?
If there's a mistake, a mutation takes place, you may get a case where that happens, and we call it a mutation, and this will give rise to something else, and we can talk about that when we talk about the mutations some other time. But generally, adenine combines with thymine, and cytosine combines with guanine.
All right. Thank you, Aslam.
Is that enough questions?
Yes, that's it for now.
Guys, thank you very much. I'm hoping you enjoyed what we did, and let's wait till next week. Luni.
All right. Thank you to Aslam for giving us a great lesson and for being such an awesome teacher. Mindsetters, thank you for tuning in, for asking all these questions. I hope you got all your answers and you understand fully. Remember, I did post the challenge question. I did post the notes on the page. The answer for the challenge question is posted on Facebook. So visit our page, guys, and make sure you get all that information. But from us, until next week, we shall see you. Thank you for tuning in. Goodbye.
Very seriously, start working from the beginning, and we shouldn't have any problems. Today we're looking at DNA, deoxyribose nucleic acid, and this is one part or one component of nucleic acids. We'll come to that just now. What do we want to really do today? The idea we put this on, guys, is at the end of this hour, these are the things that you need to know. So after the lesson, you need to ask yourself, do I meet these things? Okay? So the first thing we want to do is revise the structure of the cell with an emphasis on the ribosome, cytoplasm, and parts of the nucleus. This is what you would do. You will revise the cell. Go into the cell, look at the nucleus, look at the ribosome and the cytoplasm. Most importantly is the nucleus in this whole topic that we are talking about. Name the two types of nucleic acid. And again, while we're talking about this, guys, remember, look at the words. If this was a question, it said name. Did you have to discuss or explain anything? No. Name the two types. So you're going to just say what they are. We'll talk about that more later. State, again, another verb. And the nitrogenous base. These three things make up a nucleotide. But we're going to go into details just now. Then you must know the functions of DNA. Why do we have DNA in the body? And also describe DNA replication. We must also be able to talk about DNA fingerprinting somewhere in the show as well. Now, you would know that the way we've designed the program now, we have changed the way we do the lessons. So if you were in grade 11 and you were following last year, you'd know that we were doing a lot of content and so on. Now we're moving away from that. We are trying to hone in the skills, and the answers to the questions should be your content. So before we even start with today's show, we have a challenge question, which it's already, the challenge question, Luni, is already on Facebook, yes?
Yes.
It's already on the page. And I'll just read it for you again. A DNA molecule has 2,000 bases. What percentage of adenine bases is present if 400 of the bases are cytosine? That's the first part of the question. The second part of the question, what will be the percentage of guanine in this case? In the same case, what will be the percentage of guanine? Hint, remember, in DNA, the way the nitrogenous bases are combined, we have at cute girl. At adenine combines with thymine, and cytosine combines with guanine. So we're talking about at cute girl. That's how you're going to remember that A combines with T and C combines with G in DNA. So you think about that, you answer that question, and try and show working when you're answering the question so it makes sense to you. Now let's look at what this section is all about. Summary of the work that we're going to do. What are we talking about? DNA. But in this DNA, we don't only look at DNA itself, but today we're looking at DNA, remember that. But on the whole, the section deals with nucleic acids. These are molecules that are found in the nucleus and the cytoplasm. What are these two nucleic acids that we are talking about? We talk about DNA, deoxyribose nucleic acid, saying state where it occurs. So there's no discussion, you just got to give the location. Describe, I'm so glad, and guys, I must tell you, this is not even planned, and I'm so glad that it was put this way, because these verbs are the most important thing in the life science, the verbs in the question paper I'm talking about. Just today I was at a roadshow and we were talking about the action verbs, but we'll talk as we go along. Look at this one, it says describe now. Describe the history of the discovery of the DNA molecule, Watson, Crick, Franklin story. What happened? How was this molecule discovered, etc.? Again, describe the structure and name. Look at that now. In the one sentence, you're getting two action verbs. Describe the structure. In other words, you're going to tell us what the structure is, and name the three components of a DNA nucleotide, a big word there as well. We'll talk about that a little bit later. As follows. State the functions of DNA and describe DNA replication. The three components missing here for some reason is the phosphate, the deoxyribose sugar,
What's up, Matrix? It's finally your time to shine. You hang out with Looney, and then he said the mad guy is back. Aslam, how are you?
Ah, Looney, what's happening? They call you Looney, but I'm the mad guy.
Yeah, you know,
That's the problem, eh?
this thing. What are we doing for the Great South today?
Today we're doing the magic stuff. The magic stuff, DNA. That thing that gives you your color of your eyes and the shape of your eyebrows and the rest of it.
Everything. Okay. Great Tales, I hope you guys are excited. Just before we start, happy birthday to Llewellyn, who didn't tell us it was his birthday today. Happy birthday to you. Hope you enjoy the rest of your day. And mindset is to get a hold of us, Facebook and Twitter, @LearnExtra. And you can visit our website, learn.mindset.co.za. And then Aslam will give you a great lesson.
Wow, Looney, you're learning to talk a bit faster.
You know, I'm trying. I'm trying. It's a new year. It's a new year.
Hi, guys, and welcome 2014 grade 12s. This is your big year, the year where the rest of your life may be decided in terms of a career or job or whatever else that you're thinking about doing.